Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Okay, let's unpack this. Imagine everything you thought you knew about how a key brain signal works was missing a whole, like, star-shaped piece of the puzzle. For decades, we believed a crucial brain chemical called norepinephrine, or N, famous for sharpening our attention and kicking our arousal into gear, acted directly neuron to neuron. But this deep dive reveals something, well, truly surprising. N might be acting more like a whisper to a trusted go-between, rather than a shout to the direct recipient. We're diving into a groundbreaking paper from Science, published in 2025, thrillingly titled, Norepinephrine Signals Through Astrocytes to Modulate Synapses. Our mission today is to understand how astrocytes, these incredible star-shaped brain cells, aren't just passive supporters, but are actually obligatory intermediaries for norepinephrine's effects. This completely reshapes our view of brain communication and, excitingly, opens up brand new therapeutic avenues.
Yeah, what's truly fascinating here is how this research directly challenges a long-held dogma in neuroscience. I mean, it really is. It's a paradigm shift, moving us beyond a purely neuron-centric view of how our brains actually tune in to the world around us. It's like finding out the stage crew has been directing the show all along.
That's a perfect analogy. So, to set the stage, let's talk about the classic textbook explanation. For years, we've understood that norepinephrine, NEE, is this powerful neuromodulator. It's released by a tiny but mighty area deep in the brainstem called the locus coeruleus. NE is like central to our ability to pay attention, to learn new things, and to regulate our overall arousal levels. And the prevailing wisdom has always been that it achieves this by binding directly to specific protein receptors on the surface of neurons, initiating a cascade of changes. That's been the established understanding, right?
Precisely. That was the prevailing view, and, for good reason, it's how many current drugs targeting ND pathways operate. However, in parallel, we've long known that astrocytes, those critical star-shaped glial cells that intimately surround nearly every synapse in our brains, also carry a particular type of receiver called A1 adrenergic receptors, and we'd seen them show these intriguing surges in calcium, like internal flashes of light whenever any levels would rise in the brain.
Okay, so on one hand, the textbook said NE works directly on neurons.
Okay.
On the other hand, these astrocytes were clearly responding to NE. For years, was that just considered like a curious side observation, or did researchers suspect it was more significant? Was that a dismissed detail or an open question just waiting for its moment?
That's an important distinction. For a long time, researchers weren't entirely sure if those astrocyte calcium flashes actually mattered for how synapses function, or if they were just the astrocyte sort of eavesdropping on neuronal conversations without actively participating. There were early hints from lab studies suggesting astrocytes could secrete other signaling molecules like ATP or D-serine after NE exposure, but the evidence from live animals was sparse and sometimes even, well, contradictory. So it wasn't exactly dismissed no, but definitely left a huge frustrating gap between what we observed at a cellular level and how any impacts entire brain circuits. Was it just background noise, or were astrocytes actively shaping the neural landscape?
That truly sounds like an open puzzle. Were astrocytes merely passive observers, just listening in on any signals, or were they crucial obligatory intermediaries? Meaning, were they essential messengers that translate these brief bursts of NE into lasting changes in how synapses, those tiny connection points where neurons communicate, actually function? And solving this puzzle matters profoundly. I mean, astrocytes vastly outnumber the very specific tiny spots where NE is released onto neurons. If they truly relay NE, it fundamentally means our classical neuron-centric view of attention and memory needs a serious update, potentially revealing entirely new drug targets for common conditions like ADHD or even stress and PTSD.
Exactly, a really serious update.
So how did this team of researchers finally crack this long-standing mystery? They homed in on a critical conceptual gap, right? How could widespread NE signals, diffused across the brain, gain such specific precision at individual synapses? Classical models, with NE acting directly on neurons, just couldn't quite explain that level of fine-tuning. Here's where it gets really interesting. They began by recognizing that astrocytes are actually the brain's primary reservoir for those I1 adrenergic receptors we just talked about. And since astrocytes physically wrap around thousands of synapses, they could theoretically take those diffuse NE signals and focus them into incredibly precise nanometer-scale chemical changes, like little focusing lenses.
Exactly like focusing lenses. And if we connect this to the bigger picture, demonstrating this kind of relay mechanism wouldn't just refine our understanding of neuromodulation, it would fundamentally redefine it. It also strongly highlights glial cells like astrocytes as potential new therapeutic targets. Consider that many adrenergic drugs used today have broad side effects precisely because they hit neuronal receptors throughout the brain. Astrocyte-targeted strategies might offer a pathway to much more precise interventions, potentially avoiding those widespread issues. It's an elegant solution, really, addresses both a fundamental scientific question and a practical therapeutic challenge.
Elegant indeed. And it makes you wonder why this particular puzzle remained unsolved for so long. Was there a conceptual blind spot, maybe, or just a lack of the right tools? Regardless, the paper lays out 4 key innovations that allow them to finally make this breakthrough. Let's walk through them, almost like following a detective story.
Absolutely. Each step was a testament to combining different cutting-edge techniques and overcoming significant technical hurdles. It wasn't easy.
So the first major hurdle was simply being able to observe this process accurately. How do you observe norepinephrine's subtle whisper to an astrocyte when your existing tools are essentially designed for much louder neuron-to-neuron conversations?
That's exactly right. Imagine trying to listen to one specific conversation in a crowded, noisy room without the right microphone. It's kind of like that. Previous NES sensors primarily picked up neuronal responses. So to solve this, they brilliantly engineered a custom astrocyte-specific NE sensor, which they called GA-BAN GFAP. This allowed them to use a technique called two-photon imaging, which is like using a super-focused laser microscope to directly visualize any dynamics only in astrocytes in the live mouse cortex. It was a crucial first step, letting them isolate and observe glial responses like never before. This was a critical technological leap, allowing them to finally see the whisper.
A truly critical leap indeed. It's like finally putting on the right glasses to see something that's always been there but was just, blurry. So once they could see it, the next logical step was to prove cause and effect, right?
Yeah.
How did they establish that NE was causing these astrocyte responses, not just being correlated with them?
Exactly. This was their second major innovation. Seeing is one thing, proving causality is another. They paired their new astrocyte sensor with optogenetic activation of the locus coeruleus axons. Optogenetics, for those unfamiliar, is a powerful technique that uses light to precisely control genetically modified cells. By shining light, they could precisely trigger the release of physiological levels of NE and, for the first time, establish a causal link between that NE release and the subsequent astrocytic calcium waves. This wasn't just correlation, it was direct evidence that NE was initiating the astrocyte's response. They could essentially flick a switch and see the astrocytes light up in real time. Pretty cool.
So, okay, they've established that NE directly causes these astrocyte reactions. It's not just a bystander effect. But the next, perhaps even more crucial question is, if astrocytes are reacting, what do they do with that information? What's the actual signal they send out to influence the neurons? What's the secret messenger that the astrocytes dispatch?
Right. This is where the detailed molecular work comes in, our third innovation, and it's a real game changer. They suspected that astrocytes might be releasing ATP, a molecule often thought of as cellular energy, but which also acts as a potent signaling molecule in the brain. To prove this, they created an inducible, astrocyte-restricted knockout of a gene called VNUT. VNUT stands for vesicular nucleotide transporter. Think of VNUT as the tiny delivery truck inside astrocytes that loads ATP into little packets for release. By disabling VNUT specifically in astrocytes, they eliminated the astrocyte's ability to release ATP, crucially without affecting neuronal signaling. That specificity was key.
And what did they find when they blocked that ATP release from the astrocytes? Did the NE signal still get through to the neurons in the same way?
Well, what they found was truly compelling, and it completed a critical part of the puzzle. They discovered that astrocyte-derived ATP, once released, is then rapidly converted into another signaling molecule called adenosine. And this adenosine then binds to specific A1 receptors on nearby neurons, which in turn depresses excitatory postsynaptic currents. For our listeners, that means it essentially puts a soft brake on how easily those neurons get excited and send out their own signals. It sort of dials down the volume. This three-step process, and talking to astrocyte A1 receptors, leading to ATP-adenosine release from the astrocyte, which then influences the excitability of the nearby neuron, was a major discovery. It connected the dots from a widespread general signal to a precise, localized synaptic effect. It's like the dispatcher receiving the citywide alert, then sending out a specific message to a particular street to dim the lights.
Wow.
Oh,
That's
yeah.
intricate. It's all incredibly fascinating at this cellular molecular level, but what does it mean for the whole animal? Does this pathway actually affect behavior like how a mouse learns or pays attention? That's the ultimate test, right?
That's exactly the ultimate test, and it brings us to their fourth innovation, real-world impact. They used a sophisticated technique called closed-loop chemogenetics to precisely disrupt either the astrocytic I1 adrenergic receptors, those NE receivers on the astrocytes, or the VNUT pathway, which we just discussed, in behaving mice. Chemogenetics is another powerful tool that allows researchers to control genetically modified cells using designer drugs. It gives you really fine control. Critically, they observed that disrupting these astrocytic components impaired the NE-dependent enhancement of sensory discrimination in the mice.
So wait, it directly impacted their ability to distinguish subtle sensory information. That's a direct behavioral link.
Precisely. This is powerful behavioral validation. It directly connects this previously unknown cellular pathway NE to the astrocyte's I1 receptor, leading to adenosine release, which then influences synaptic function, to how animals process information and learn. It's a pathway that classical neuron-focused approaches had completely missed. It truly demonstrates that astrocytes are not just support staff, but active participants in shaping our perception. And it's important to note, this wasn't an isolated finding. 2 other independent studies, also published in Science alongside this one, provided corroborating evidence for astroglial involvement in NE or even tyramine signaling and their impact on purinergic signaling. It reinforces this new model of neural network function. It's a clear signal that the field is moving in this direction, which is exciting.
So, okay, let's recap the whole journey then. Norepinephrine talks to astrocytes.
Via those alpha-1 receptors.
Right. Astrocytes then release a specific signal ATP, quickly converted to adenosine.
Exactly.
And that signal then fine-tunes how neurons communicate, how sensitive they are, and ultimately how you perceive the world and focus. Pretty mind-blowing, right?
It truly is. It makes you wonder what else in our own brains we've been oversimplifying. It's like finding out the road crew isn't just patching potholes, but they're also subtly directing traffic and changing the speed limit on the fly, depending on conditions.
Exactly. So this really shifts our understanding of the brain's fundamental wiring. What does this all mean for the bigger picture of neuroscience and maybe for potential applications in treating brain disorders?
Well, the work profoundly elevates astrocytes. They're no longer just passive sensors that listen into any signals. They're now active, even obligatory modulators. This really forces a shift from a purely neuron-centric view to what we call tripartite models of noradrenergic signaling, where the synapse isn't just 2 neurons talking, but also an astrocyte listening, processing, and actively influencing that conversation. It's a far richer and more complex picture.
So it's not just the neurons, it's the neurons and their star-shaped support system actively working together, like a truly integrated team, a 3-way conversation.
Exactly, a 3-way conversation. And beyond the theoretical shift, the new tools developed here are huge, like the GRABNY GFAP sensor for seeing NE in astrocytes, and the astrocyte-specific VNUT knockout mouse for manipulating their signaling. These are incredibly valuable. They expand the glial targeted toolkit for researchers and are likely to be widely adopted in studies of other neuromodulators, allowing us to ask similar questions in entirely new contexts. It's like being given a brand new set of advanced magnifying glasses and scalpels for brain research.
That makes sense. And on the application side, this isn't just about understanding the brain, is it? It sounds like it has direct implications for drug discovery.
Absolutely. By pinpointing the ATP to adenosine step, this study identifies VNUT and other related enzymes as really tractable drug targets, potential targets we didn't even consider before. This offers the promise of tuning any effects with much greater spatial precision than current broad-acting drugs. Think about the implications for attention disorders like ADHD or even conditions related to stress resilience and PTSD. We might be able to develop therapies that specifically target these astrocytic pathways to fine-tune NE signaling, potentially avoiding the widespread side effects like sleep disturbances, anxiety, or appetite changes of drugs that hit neuronal receptors throughout the brain. Instead of a general alert, we're talking about a surgical strike, much more targeted.
It sounds like a whole new frontier for drug discovery, potentially leading to much more refined and targeted treatments.
It truly is. Of course, the authors are clear about limitations too, as good science always is. You have to be. This pathway was primarily demonstrated in the cortex. We don't yet know if the exact same relay operates in deeper subcortical regions of the brain or under conditions of chronic stress, which could alter the dynamics significantly. And deleting VNUT might trigger compensatory mechanisms over time that these acute experiments didn't capture. Science is always building on what came before, asking the next question.
Right. There are always more questions. But despite those open questions, this feels like more than just an incremental step in our knowledge. It feels like a genuine shift, doesn't it?
It absolutely represents a true paradigm adjustment. It integrates glia, these previously underappreciated cells, into the mainstream theories of neuromodulation. And it equips the field with practical genetic and optical tools to push forward. Future clinical translation will depend on developing those astrocyte-selective modulators and validating this pathway in actual human disease models. It's exciting work ahead.
And it's also worth appreciating the incredible collaboration behind this. This wasn't just one lab. This was a multi-center transcontinental effort, bringing together expertise in molecular engineering, deep brain optics, and systems behavior. It's a testament to how diverse specialties can accelerate discovery when they truly come together.
Absolutely. A real team effort.
So the next time you're trying to focus or learn something new, remember that it's not just neurons at work. Those star-shaped astrocytes might be quietly, intricately orchestrating the signals, acting as the brain's hidden intermediaries, fine-tuning your very perception of the world.
It certainly raises a provocative thought for you to mull over. If norepinephrine uses astrocytes as intermediaries, what other crucial neuromodulators, perhaps serotonin or dopamine, might be secretly working through these fascinating glial cells? And how might unlocking these hidden puppet masters reshape our therapies for neurological disorders previously thought to be purely neuronal? That's something to think about.
A truly fascinating question to ponder. We hope this deep dive into astrocytes and norepinephrine has given you some surprising facts and a whole new perspective on how your brain works.